All right, good afternoon, everybody. Welcome, welcome, welcome. Just to be clear, I am okay with everyone choosing if they want to wear masks or not. I'm generally distanced from all of you, and in this community at OWU, where virtually everyone's vaccinated, I feel pretty safe, so I'm not going to wear one. Especially when I'm teaching, it's actually kind of nice not to wear one. But of course, do what makes you feel comfortable, what you want to do. That's great. It is super weird to see everyone's faces. I will acknowledge that. There's like, so even people whose faces like I know, but I haven't seen in a long time, it just throws you off because you kind of like fill out the rest of people's face and you kind of have an idea what that's going to look like. And then they take their mask off and you're like, oh, that's weird. So you can get over that. This is what my face looks like. We'll get used to seeing each other again. So our last class before spring break is today. We've got an agenda, kind of a more traditional class structure agenda for you after Monday's experiment. So let me just back up for a second. Monday's experiment, I felt like at the end of the day, Princeton and I debriefed a little bit afterwards, had just the right amount of chaos. When we do something like that, there's really not a way to avoid chaos that goes with doing that. It's kind of an accurate representation of how running an experiment is sometimes as well. I really want to say I appreciate all of you that like stepped up, that participated in measuring the animals and doing the cleaning performance, that acted as a lizard catcher, all of that. It was really great to see that. We're going to take all of those data and kind of continue that project and analyze it and look back on your predictions based on the influence of size and temperature after spring break. So we've got a few things that I want to do today before spring break. I kind of had today planned out already. And then we've got spring break and some other stuff. I'll review the schedule in a second. I just want to let you know that we're not done with that yet. We're just going to kind of put it on the back burner for a couple weeks and get back to it. Having said all that, any questions about what we did yesterday and what we've got going on moving forward? All right, there's Sam. We can start now. Sam's here. Sorry. I'm just kidding, Sam. I'm just messing with you. Okay, so today's agenda looks like this. First, I have herpetology in the news for you. An interesting story I came across recently. Then for today's class, you were writing one of two papers on lizard performance. And we're going to work in pairs in trade, teach each other about those papers. And then I have kind of a chalk talk about those two papers, some main concepts I want you to take away. I'm going to do a kind of impromptu improv lecture with those materials. And then, my friends, you get to have me do another seminar with you. So I am going, I'll explain the context of it actually when we get there instead of now, but I'm going to give a talk today that links primarily about thermal physiology, which is the next kind of topic we're covering in class. This next unit focuses on functional morphology. We started yesterday, and then thermal biology as well. And then we're going to introduce that today with a kind of seminar style talk. Then we'll wrap things up. I've got your quizzes graded and ready to get back to you. I'll share that with you, and then wrap up and some reminders before I don't see you for a week and a half, which will be sad, but we can make it. Questions, comments, concerns? All right, herpetology in the news. So since you're not going to have a class program over break, I'm not going to include questions on this on your lecture, your quizzes or exams as I normally do. It's more something I came across that is just interesting. So this is a CT scan of a lizard, and it is a species called the curly-tailed lizard. These guys are invasive in Florida and actually causing a lot of problems for native lizard species there. And that's actually why I came across this article, because I was looking up stuff about invasive lizards. We talked about Podarcis moralis in Cincinnati last week. And so I was just kind of thinking about this, and I came across this crazy article. This is a CT scan. You see the animal skeleton. This brown bolus right here, this is poop. This lizard set the record, the known record for vertebrates in the ratio of the size of a fecal pellet to the size of the body. So this poor guy's digestive system was completely impacted. You can see the size of this thing that leaves just this tiny little bit here for all his organs. Lungs, heart, liver, all of that is compressed up in this little area. The rest of this is one giant turd. The poor lizard actually was incapable of passing that. They had to euthanize this animal. But what's interesting is they actually found this guy. So we talked about invasive lizards, but also urban lizards, right? These guys were living behind a pizzeria around a dumpster where they were eating invertebrates and like scraps of food and stuff next to a grease bin. And so I'll just read this quote to you. It says, feasting at Cocoa Beach Pizza Parlor grease bin gave one northern curly-tailed lizard a record-breaking case of constipation. Mouthfuls of insects and a knoll and greasy sand congealed an unpassable glob of poop, nearly 80% of the animal's body mass, the largest known feces to body mass ratio recorded in a living animal. So take a second, do some quick math, calculate 80% of your mass, and figure out how big that would be relative to you. That's a lot of poop. Interestingly, these guys are super successful invaders, but we can see here that under the wrong circumstances, greasy sand can do them detriment. You just did the math there, Sid? Yeah, that's a lot, right? Yeah, okay. So that's it. Again, I'm not going to include this one. And the reason I'm not going to include it is because normally I put links to these in your program. We don't have a program, so just wanted to share this interesting piece of information with you. I know you were all wondering what vertebrate has the record for the largest feces to body mass ratio. Curly-tailed lizard. And my slides are screwed up, the wrong order. Stretch break, guys. Good job, everybody. You're done. We'll get to that. All right, so I want to start in talking about these papers. You all read one of these two papers, and your assignment was basically to prepare to teach someone about it. The first paper is looking at across species, so an interspecific comparison of performance in different situations for lizards in the family Lizardidae. And the second was an intraspecific paper looking at variation in morphology in relation to performance in anoles that are colonizing urban habitats. So what I want to do today is just an activity where I'm going to pair you up based on who read which paper. And so basically the person next to you on this list is the person who you're going to partner with. And we're going to spend 5 minutes each way, so 10 minutes total, talking about the paper. So it's going to be helpful to have a copy of it pulled up as a PDF or printed or whatever. And I'm going to challenge you. This is the challenge, is you're going to talk about just one of the papers for 5 whole minutes. Even if you think you're at the end, you've explained everything, focus on that one paper. And this goes both ways. Both the person explaining and the person receiving information need to participate. If you're receiving information, you need to ask questions. You need to think about it. You could do anything for 5 minutes. So focus, you're on task, 5 minutes just talking about that one paper, not diverging into anything else, not diverging the other paper, and then we'll switch and go back the other way. Let me see, I know Nebraska's not here. So Chloe, you need a partner. Is anyone else missing today? And McKenna, yeah, I knew McKenna, she's actually not on the list. What's that?
I can't see like the bottom half of the board, sorry.
Oh, you can't see which?
Taryn's not here.
Oh yeah, Taryn's missing. So, okay, so that's perfect. So Abby and Chloe can be partners. Does that work? Oh no, wait, this is Abby. I apologize. Abby with whether or not has an E, I get mixed up. This is you, Abby. Okay, so that's Abby and Chloe can be partners. That's perfect because Taryn's not here. Boom, that works out great. Look at that. We put our heads together, we come up with a solution to what seemed like an unsolvable problem. We can do anything. Yeah, we're impressive. Okay, go ahead, get with your partner and take 5 minutes to go ahead and get started. Those of you watching the recording, I'm going to pause the recording here. There's no point in attending 10 minutes recording everyone talking. All right, let's go ahead and bring it back here if we could. I have to say that I'm absolutely thrilled with what I saw when I heard just now. That was great. You were on your focus, like everyone had like these really nice summaries and information about the papers in different ways. It took different formats, but that's great. That's exactly what I was hoping we'd see, is that everyone kind of does it the way that they think is best, and that's going to be all different ways, and that's going to be all good ways, right? So yeah, we'll do more activities like this with reading. What I like about this too is it allows us to kind of double up on the literature. Without assigning you 2 papers, now you have a pretty good kind of idea of the fundamentals of a couple of papers that are relevant to course topics. So awesome work, everybody. I'm really thrilled with that. Just as a logistical issue, remember that the activity was 6 class points, so you'll get those in Blackboard soon. But all of you here have earned the points for that. What I'd like to do is, I have 4 topics as I read these papers, like ideas that I just want to take some time to unpack and make sure are clear. So I'm going to do a little chalk talk here. If I had any chalk, it'd be helpful. Here we go. The first thing is that one of the papers, I'm going to spell this wrong. Would someone just confirm that I spelled this right? Van Hooydonk?
Yeah, that's right.
That's okay? Okay. And Winchell. I'll just throw in a little plug here. Kristen Winchell. Oh yeah, you go back to your seat. Sorry, I should have said that before. You don't have to be with those partners anymore. Unless you made a new friend and you want to sit with them for the rest of class, that's great too. It would be so great if like people got assigned random partners in my class and they ended up like dating or something. If that happens from this activity, will you guys just let me know? Like I don't need to know details, but if I could somehow facilitate like a budding romance, that would make me incredibly happy. Oh, that was in astronomy. Yeah, I didn't talk to her. That was like the opposite of what I mean here. No, when I was an undergraduate, I had an astronomy class, and then these tables set up. I don't know why I'm telling this story. I'll just make the quick version. There was like 2 students at each table, right, around the room. And every day in class, I sat next to this other student on whom I had a huge crush. And I just never said anything to her until like the last class or something. I was like, hi, and she's like, hi. It was super, yeah. I blew that. But I hope all of you have better social skills than me, so maybe, who knows. Yeah, that's a different story. All right. I'll tell you that story another time. All right, so we read 2 papers today, Van Hooydonk and Winchell et al. I want to throw in a quick plug here. Kristen Winchell, the lead author of this paper. Princeton, do you want to tell us just a little bit about Kristen, how awesome she is?
Sure. Well, Kristen's pretty awesome. So for our experiment that Dr. Gangloff and I are doing right now, we actually reached out to her because she's using a method that we have no experience with. Basically, it's a method to like quantify shape of an object. And so basically we like reached out to her and was like, hey, help, please. And she was like, oh yeah, this is how you do it. And we're going to set up this Zoom meeting and this person's going to teach you how to do it. And you're going to be great. And we're also going to share with you our entire code and you're all set. And so basically we reached out to her and she did all that and she's a lovely person. And yeah, and we would not be doing this experiment without her help. So she is super nice.
Agree. There's our love letter to Kristen Winchell. Okay, so you read 2 papers for today. What I want to emphasize first is important difference. This Van Hooydonk et al. Paper is an inter-specific comparison. And this is a really important distinction to make between these 2 studies. The Winchell et al. Paper is intraspecific, which means that for the first paper here, what they did was they took averages across a whole bunch of different species in the family, and their unit of, their experimental unit is a species. So their question is about how do these species differ in their performance, in their relationship between morphology and performance. And they had what, 13 species, 11 or 13, I don't remember exactly. So when you, 13, yeah. So when you see those plots, each of those dots represent a whole lizard species. And to get that value, they took the average of like a whole bunch of individuals, right? So imagine a study that's comparing, let's just take it to our level, the sprint speed among great apes. And so we would have one group would be humans, Homo sapiens, another would be chimpanzees, another would be gorillas, and another orangutan, to say, for example. We're going to measure our femur length versus our sprint speed. Each of those values in that case is the average across the entire species, right? So think about that for humans. If you took the average sprint speed for a human, it may or may not be representative of an individual, right? Because there's going to be a lot of people that are faster and a lot of people that are slower. But they're distilling all that down to the species level to ask questions about how species differ, and presumably those differences are due to evolutionary processes. And quite assuredly, that's how speciation happens, right? We have these different species, and they have these different traits and these different relationship, in this case, between morphology and performance. And so their question is looking at differences among species. And I just want to emphasize that the values they're using for that analysis are these averages among all these different individuals. The Winchell et al. Paper, on the other hand, looks at intraspecific variation. And for me, this is, oh, actually, let me back up for 1 second before I talk about this. This is one other point I want to make. Using these interspecific comparisons in looking at things in this context, this is what we call a comparative study. Now, the word comparative can have many different meanings, but specifically in evolutionary studies, a comparative study looks at differences in traits among different taxa. In this case, different species. You could do a comparison and compare, I don't know, something else among different families within a phylum or something like that. Most often it's species though. And so this is what we call a comparative analysis. Sure. A comparative analysis compares traits among different taxonomic units. In this case, it's comparing traits for performance among different species.
It has to be the same trait, correct?
You have to compare the same trait. You can compare multiple traits. You can compare leg size and sprint speed and climbing speed and all these things, but you can't compare leg size in one species to sprint speed in another species because it's two different things, two different units, two different scales, all of that. So a comparative study is specifically looking at variation among clades, right? In this case, species. An intraspecific study, on the other hand, is probably something that is a little more familiar to all of us. And it's actually quite more familiar to me in my research, is almost always intraspecific. And this looks for variation among individuals. So in this case, the experimental unit is an individual. One little lizard, right? And we look at the morphology of that single individual lizard, we look at the performance of that lizard, and then we look for a relationship between that morphology and performance. So for humans, the equivalent of this in humans would be, okay, let's have everyone in this room run the 100-meter dash, and then we're going to measure the femur length of everyone in this room, and we're going to see if there's a relationship between the femur length of people in this room and how fast they can run, right? That would be intraspecific because we're all the same species, and we're linking up those measures on a single individual. And what's important for this is that we have measurements of the different traits on the same individual. So we need to have Carissa's femur length and her sprint speed together. It's not really useful, although in some contexts you can do that, but it's not as useful, I'll say, to have this class all run the 100-meter dash, this class we're going to measure your femur length, and then we're going to see what we can find. Well, we don't know how fast anyone is for whom we have femur length, nor do we know the femur length of anyone that we have sprint speed for. And so there's really not much you can do with that. The key here is that you have to measure multiple traits on the same individual to look at those relationships. So we have intraspecific and interspecific studies. I want to make sure that's clear because it's really important, determines the kind of conclusions you can draw from those studies. I'm going to pause here and just make sure those ideas are clear, see what questions you have, and move to topic number 2. And if someone mind taking a picture of this notes, and you can just send it to me later. I just want to make sure I have these to make sure I know what we covered, and then we can share it with anyone that's not here or charge them for it. Let's see. How much? Dang, Sans. All right. Yeah, this. Yeah. Yeah, this discussion probably just cost you all $20. Yeah. Sorry to say. Yeah. If it makes you feel any better, I just got a new repayment plan for my student loans, and I'm scheduled to have them finished being repaid in 2045. So yeah, I feel the burn. Yeah, so good luck with that. All right. I went to an expensive school for undergraduate and for my master's, and I didn't really have any recognition at the time that I would have to. Anyway. It would have been worth it if I had talked to the student sitting next to me in my astronomy class that I had a crush on. I would have different feelings about that. Don't tell my wife I said that. All right, questions on inter versus intraspecific studies. Okay, I'm gonna erase. Prince, do you have another here? Would you mind erasing from that end? Nice catch. All right, so topic number 2 is going to be, there's 2 important differences, I think, in these 2 studies about how they handled temperature. And so Someone who read the Van Hooydonk et al. Paper, I gotta spell that again now.
Whoops, sorry.
Oh, that's okay. I was actually just thinking that I wanted to say that after we erased it. I'm just gonna say the Van Hooydonk. How did they handle temperature in the Van Hooydonk paper? Say again?
35 for everything.
Mm-hmm.
Maybe like 1 or 2, 38.
Yeah, so they controlled the temperature. Most at 35, and a couple species at, what, 38 you said? 38 C. In other words, they determined the temperature that the lizards were at before they sprinted them, the idea being that these were the optimal temperatures for performance. I'm just going to call that the optimal temperature. And this is sometimes abbreviated as T-opt. And this is actually going to come up in the talk I'm going to give here and after our break, this idea of T-opt. But the optimal temperature for performance, they knew what that was generally for the species, so they did all the sprint speeds at that temperature. How did the Winchell et al. Study control for temperature? Sid, go ahead.
I feel like this is probably not right, but I don't know that I remember reading about it. Like, I don't know that they really did. I'm sure they had a control temperature, but they didn't really talk about it, I don't think.
They did mention a little bit about it.
If I'm
Yeah.
missing that.
Anyone want to jump in? It's a pretty obscure line for the methods. So unless you think about temperature in relation to performance all the time, like I do, you might just gloss over it. Yeah, look.
It says that they set the track at 37 degrees melt.
Yeah, that's the incline, right?
Yeah, that's
right,
why
right.
I was like,
Yeah.
wait, what?
Anybody? Yeah, go ahead.
They recorded the body temperature right at the start of the performance test.
Good. Yep. So they just recorded the body temp. So they kept the animals, I think they set an ambient temperature wherever they did the experiment. And before they did it, they just recorded the body temperature and then noted that. And then they recorded the temperature and then controlled for it statistically. And so in this top study, they don't need to put temperature in their statistical models because the temperature was the same for all of the animals, basically. I mean, it was slightly different, but that's based on species differences. So we could say the temperature was the same in the sense that it was the optimal temperature for all the animals. This study didn't control the temperature, just had the lizards out. They would record the body temperature and then sprint them. And they took that information and put it in their statistical tests. That way, if there was an influence of body temperature, which there was, then they could control for it mathematically in their statistical models. Which way is better?
I feel like both ways are okay. If you control for it,
Mhm.
I like the top one more because I'm not taking things for granted.
Yeah. Good. Other thoughts on that?
Yeah, I don't really see the point of, I mean, I'm sure there is a point, but like, trying to statistically control for something when you could just do that yourself
Mhm.
and not have to worry about any margin of error or anything that could go wrong with the math or some sort of data point that would throw off potentially your entire result with that.
Yeah, it's a much cleaner experiment when you have everything done at the same conditions, right? Yeah. Good.
So I'm actually in psychological research methods.
Yes. Tell us. Yes.
One thing about the second method would be that it might be more generalizable to lizards that are actually living out in the wild because they have different body temperatures. You know, they're not completely controlled for all the time.
Mm-hmm. Good. Yeah, you could make the argument that like that's a more natural body temperature for them to be at to perform those tests. And that's a great argument. The thing, or Ethan, go ahead.
I don't know if you know this based on the statistical test they use, but would that lower the overall confidence in the results, the bottom method?
It shouldn't. It depends. One thing I wish they had done and that they didn't do is record the range of temperatures that they recorded. Like they said, we recorded the temperatures and we analyzed them, but it doesn't say anywhere in the paper, at least that I saw, they range from 15 degrees to 35 degrees C. I would tend to agree with both of you that there's different circumstances that in which either method could be better. What you need to watch for, though, is in this paper, let's say that there was a day when they measured a whole bunch of urban anoles, and there was another day where they measured a bunch of forest anoles, right? And they're comparing them. What if one of those days was warmer and one of those days was cooler? And so you confound, they measure all of the urban anoles on a warm day, all of the forest anoles on a cool day, and then you're confounding your question of interest. Is there a difference between urban and forest anoles with temperature? What you're not really interested in here, you just want to control. So yeah, you go either way. And the important thing in a study is that you report exactly what you did so that anyone reading it can make an inference and kind of know what that is. And either say, yeah, that makes sense, or I'm not sure if that's really the best way to do this, but as long as you know what the this is and you can understand it, then ultimately that's what's going to be important. Every study design is going to have something that is really works well and things that maybe aren't as perfect just because of logistical constraints. But in my opinion, the important thing is to just report it. So cool, good. Questions on that? Now we know that temperature. Princeton, want to erase that? Oh yeah, picture. It's $20. We know that temperature is going to be important for all aspects of ectotherm performance. That's actually what we're going to talk about in a little bit. I've got two more quick ideas I want to run by you here and make sure we understand. Yep,
The names.
yeah, you can leave the names. Another vocabulary word I want to talk about is this word that came up in the Van Hooydonk paper, something called a polytomy. A polytomy is basically a phylogenetic question mark. It's basically a situation where we're like, I don't really know what happened. So we have a phylogeny that looks like this. Let's say there's our root. We have a branch here. We have a couple other species over here. We have species 1, species 2. I'm being really creative here. This branches off. But then we have a situation like this. We have species 3, species 4, and species 5, right? So you can see here that species 1 and 2 are sister taxa to each other. They form the outgroup to this group, species 3, 4, and 5. Now notice here that we don't have a bifurcating line. Instead, we have this line splits off 3 ways, right? This, my friends, is a polytomy. How you have all 3 of these lines having equal evolutionary relationships. This is really rare, almost incredibly rare to happen in nature where you have this actual situation. So what this basically means is that we don't know that relationship. That's all it means here is like, we're not sure if species 5 is more closely related to species 4 than it is to species 3, or maybe 5 and 3 are more closely related to each other than they are 4. We just don't know. So we're going to make our tree like this and just go with it because we're not sure of that relationship. Go ahead, Sid.
So I was just gonna ask, is actually this particular phenomenon extend to the entire chart, or it's just never happened?
Biologically?
Yeah.
So in an evolutionary sense, it would be exceedingly unusual circumstances in which one species would split into 3 species all equally related to each other. I guess if there was some example of like polyploidy happening or hybrid, something really whack-a-madoodle.
I'm
But
more was asking about like the entirety of a tree, like where we don't know what split off into what.
yeah, oh yeah, that happens all the time. I mean, they're revising even like the relationship among like organisms at high levels of our understanding of how things are related, yeah. But it's pretty common, and this paper is 20 years old or so they didn't have as much molecular evidence about the relationship of these groups. So we have something called a polytomy. We'll come back to that. I mean, this idea will come up in class at different points. But basically, the takeaway is we just don't have enough information to know what this should look like, so we're going to draw it like this and go with it. Last concept I want to get at, and this is the one that I've been waiting for because I'm really excited to talk about statistics. Some of you have probably seen this. I want to make sure that everyone understands what they mean when they say residual variation, or for example, residual hind limb length or residual speed or something like that, right? So let's say that we have a data set, got a y-axis here, an x-axis here. Our x-axis is going to be body size. So I'm going to put snout vent length. Our y-axis here is going to be, let's just say leg length, for example. So what do we expect this relationship to look like? Just off the top of your head, what do you think it'll look like?
Should be
Positive, yeah. Why positive?
because as you increase increase your overall size, one would assume that your bones would grow as well.
Yeah, generally the bigger the individual, the bigger the legs you have, right? So I'm gonna add some data points to this. We might have one here, 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 here. Something that looks like this, right? So each of these is an individual for which we have snout vent length and a body length, right? Overall, very positive relationship. Generally, bigger individuals have bigger leg lengths. Sound good so far? Okay, what we're gonna do is draw a regression line through this. So this is a very commonly used statistical model where you put a line of best fit between all those dots to describe that relationship, right? And this is a statistical model. Like we can describe this with a slope and an intercept, y equals mx plus b. We can get at that relationship between snout vent length and leg length. Sound good so far? Now, it's not really that useful to compare this lizard's leg length and this lizard's leg length. Because if you look, this lizard has a leg length that's right about here, and this lizard has a leg length that's way up here, right? And we say, oh, this guy's got a much smaller leg length compared to this one. Yeah, sure, but the lizard is smaller overall too. That's like being like, well, that elephant has a bigger leg length than this seal. Well, duh, yeah, it's a lot bigger. What we can do to make that comparison is something called the residual leg length. And that is basically the distance from this point to this line, right? This point to this line, this point to this line, this point up here to this line, this point here to this line, this point here to this line. This distance, that value, that numeric value is called a residual. And what it means is how big, in this case, how big the leg length is compared to what we would expect for an animal of that size. Now, if we compare these two values, let's say we actually compare this dot right here, right? Its leg length is right about here, whereas our other guy up here, his leg length is here. But if you look at the residual leg length, the distance from here to the line, this is a pretty big distance, and it's positive. This dot is above that line. Over here, this dot is below the line, so it's negative. So this guy's got a positive residual leg length. This guy's got a negative one, meaning that for his body size, this guy's got pretty big legs. For his body size, this guy's got smaller legs. Now, the reason this is important, especially, is because let's say that you actually have two different species of lizard. So we've got a blue species and an orange species here. You can see I made these dots different color for a reason. Blue species are generally smaller than orange species. So if we ask the question, which species has longer legs? Obviously, it's going to be the orange species, right? All of their leg lengths are up here. All the blue species leg lengths are down here. However, if we wanted to ask the question, which species has longer legs relative to its body size? Well, then we have to analyze those residuals and see, and that there's no clear pattern, at least from what I drew on the board. So if we have confounding variables, in this case, size and species are confounded, one species is bigger than the other, but we want to ask the question about relative leg length, or let's say that we change this and we make this axis sprint speed. Now we can see that all of the orange lizards are faster than the blue lizards, right? But for their body size, since we know that the bigger you are, the faster you can sprint, that relationship goes away. Go ahead, Sid.
So the line of best fit you're taking out of the averages of, or line of best fit is being drawn based on all the data points.
That's correct, yep.
What, is that's establishing your baseline for how you're gonna compare the data points to each other, and that's what you just said for residual length. But what if can that be thrown off potentially by if you get like bad data?
Oh yeah,
Okay.
absolutely. If you get one guy, let's say you measure one animal and he's got a giant body, but his sprint speed is just super slow, dude, over here. Like if you get one data point over there, that's gonna change your whole line is now gonna look something like this.
How would you control and adapt for that?
It's a good question. It comes into identify. You want to jump in, Sam? What's this guy called?
You could like an outlier. Yeah,
Yeah.
an outlier. You just get rid of the outlier.
Yeah, and sometimes, you know, I mean, you don't want to just say, oh, that data point's way out there, we're going to get rid of it. What you want to do is take a close look at that data point and see, and maybe you made notes on that day when you ran that lizard, maybe you noticed that it was injured. And so it's a big animal, but it was running slow because it was injured. Well, in that case, yeah, you throw out that data point because it's affecting it's giving you bad data basically.
So this is like a pretty reliable method in comparison?
Linear regression. In many cases, it's reliable, yes. This relationship can be complicated. These things can scale like logarithmically or something different. But as a starting point, it's a great place to start. Yep, and it is useful in a ton of different contexts. We did something like this with our study, Princeton and I did, with all the body dimensions relative to body size.
Yeah, I was just asking because it seems like it would be pretty variable depending on what kind of data you have. So that's why I was asking my question.
Yep, that's a good question. Ethan, you had your hand up?
So could we consider that residual, the variance, like the baseline?
Yeah, so the variance, so that's actually the technical term is residual variance. So it's the amount of variance in your data that's not explained by the regression line. So no model is going to fit your data perfectly. Well, it can, but then you have an over-parameterized model, that's a different story. But no, for right now, no model is going to fit your data perfectly. There's always going to be residual values left over, and that residual variance is it's exactly that's the residual variance. And when you talk about like making the assumption of normal distribution of residuals, that's actually what you're referring to, is that if you took all of these data points, these residuals, and made a histogram of it, then it would fit normal distribution. And we could look at that in more detail with some computer software because I can't do that on the board.
So if, like your example there, where the baseline, the blue overall line is the baseline for in-between species, and if, say, all the orange ones were above the baseline, would it then just be the variance compared between the two species, and that's just like the overall?
Yeah, because then it can get tricky because then you're confounding species and size. And maybe what you need to test for is to make sure the two species have the same relationship between body size and sprint speed. So make sure the slope of this line is the same. Because maybe for whatever other reason, the species have a different relationship between SVL and sprint speed. And then so you need to make sure that the slopes of the line for this species and this one are the same. So let's get a little bit ahead of ourselves, but yeah, you've got to be aware of those things for sure. How many, just out of curiosity, how many of you have seen this before in other classes? Not too, about half and half. Is it useful to review this kind of stuff in class, even if you've seen it? Okay, good. Okay, cool. Let's take a break. That's great. Stats talk, done. We'll come back and I'm going to talk about thermal biology and it's going to be great. All right, let's get back to it. All right, so here's the plan. So as I mentioned, our next kind of unit in class talks about functional morphology, but also thermal biology. I'm going to give another kind of seminar style talk today in class like we did last week, which was, I have to say, was immensely helpful in me preparing for the talk at the meeting on Saturday. Yeah, Lennon, go ahead.
How'd that go?
Well, ask Athena and Ethan and Princeton how it went. No, I didn't mean my talk, just in general.
Oh, in general.
You flatter me, Ethan. It was good. Yeah, it was good. It was good. You said go ahead.
Is this going to be mandatory doing this for?
Yes. Yeah, it will be stuff that we're going to go into more depth with after break for sure. I want you to approach this the same way you approached the last one. So you're not copying every piece of information from every slide. You're taking general notes on big ideas. And what I'm keenly interested in is you taking notes and stuff you have questions about so we can talk about it and unpack it. I'm going to move through stuff pretty quick. And this is a talk I'm actually giving back in Iowa this weekend. So every year, Iowa State University, where I did my PhD, they have a spring symposium where they have people that are in the department there give talks, and they bring in outside, like one like big name biologist from outside the university to give a talk. That's not me, don't worry. And then they invite one alum back to give a talk as well. And they invited me to be the alum to come back this year. It's actually really cool because I get to go back and see some old friends. And actually, I went to this every year as a graduate student. Every year I was like sitting in the audience being like, watching these really, Iowa State has like this amazing program and some fantastic people have come back to give talks there. and like sitting in the audience being like, someday that's going to be me. And that day is Saturday. So I'm going to give a talk. It's a little bit longer than the other one. I actually haven't timed this one yet. So if it goes long, we'll be done with class no later than 4 o'clock. That much I promise. And I want to make sure we have time to give your quizzes back and stuff at the end. So I'm just going to run through it seminar style. Hang on to questions. We can talk about it at the end if we have time, if not after break. But it is all stuff that's very relevant to what we're going to talk about in class today. So it's not, I just want to make the connection explicit that the ideas I'll get to here, we're going to unpack in more detail. All right, so with that, the talk is titled Testing the Ectotherm Toolkit of Responses to Novel Environments. And so what I want to do here, let me just get there, is give you the narrative outline of what I'm going to talk about in the form, I want to tell you the story that I'm about to tell you in the form of a narrative outline. Chapter 1, the world is changing. This will affect life. Chapter 2 is we think we might understand how these changes will affect life. Chapter 3, but we don't. Chapter 4 is so we try something else. Chapter 5, and maybe it's right, question mark. And chapter 6, but probably not always, but that's okay because it keeps us biologists in jobs. So I want to start off chapter 1, the world is changing. So what I'm going to show you here is a series of slides I actually included in my PhD dissertation defense. And so this is a marina on the shore of Eagle Lake in Northern California. This is the site of, this is one of the locations where we conduct long-term field studies on garter snakes. That was part of my PhD dissertation. You can see there in 1979, it's a busy, active boat harbor. You skip ahead some years to 2006. There's still water there, but it's water levels reduced. We don't see any boats. By 2009, the water level is getting pretty low. By 2012, you see some rocks and emergent vegetation. You see this little makeshift slip out by the mouth of the marina there. By 2013, there's grass growing and there's some muddy puddles. By 2014, there's basically no water. You can see that slip just up on land. And by 2016, what was once this harbor, this marina, is now a grassy meadow. And so what I want to illustrate here is how the environment, this is one example of how environments are changing in so many ways. We're not sure exactly what caused these lake levels to drop, but it likely has something to do with changing climate regimes, changing temperature, and also due to human use of these natural resources. Now, this is just one example. These kinds of changes, as you all know, is happening all over the world. And so what a lot of us biologists do is we try to understand how these changes affect the organisms that live in these different environments. What I want to focus on today is one example of one axis of variation in the environment that humans are altering is, of course, temperature. So I'm going to focus primarily here on how ectothermic organisms respond to variation in temperature. And to start off with, I want to describe the most commonly employed method to understand this, which is called the thermal performance curve. Thermal performance curve has a body temperature along the x-axis here of an ectothermic organism, and it has some measure of performance on the y-axis. Performance can be sprint speed, like we talked about in class. It can be its ability to digest food. For smaller organisms, it could even be something like population growth. And you can see here this kind of classic shape of a thermal performance curve, where performance goes from 0 at some low temperature, increases gradually to an optimum temperature, and then decreases rather sharply as temperatures get too high and approach a thermal maximum. And we reviewed this work recently, so throughout the talk, I'm going to mention this to everyone. I also put in like all of the work that I did here is with collaborators that are also from ISU, so I like highlighted each of them with the associated studies. So in association with Brooke Brodensteiner, who's now a PhD student at Yale, we reviewed the evolution of thermal performance curves recently and how these curves might change under different conditions. Let's see here. So the curve, from this curve, we can extract important parameters, including the optimum temperature for performance. This is the one I just talked about earlier with the lizard study. We can also extract the critical thermal maximum, the performance breadth, so the range of temperatures over which an organism can achieve some proportion of its maximal performance, as well as critical thermal minimum, the temperature at which the cool temperature at which performance becomes effectively 0. Now, from a biochemistry standpoint, we have a pretty good idea of what drives the shape of a thermal performance curve from its lower limit up to the thermal optimum. This is primarily driven by thermokinetics, the interaction of subcellular components, membranes, proteins, etc. As temperature increases, the rate of reaction increases and thus increases the overall rate of performance. When we get to the other side of the thermal performance curve, there's much more uncertainty about what causes performance to crash as organisms reach high temperatures. Now, there's a couple different ideas for what might be happening here. And this has to do with how temperatures affect organisms across levels of organization. So on one hand, you see on the left here, high temperature, under the classic hypothesis, high temperature affects an organism's ability to perform because of its effect on subcellular components. So proteins begin to denature or lose their ability to interact efficiently with each other. Membranes start to lose their integrity, etc. At the same time, high temperatures might also affect an organism at the organ level system. A hypothesis recently advanced for how this might work in ectotherms posits that oxygen limitation is actually the limiting factor at high temperatures. In other words, at high temperatures, an organism's metabolism keeps increasing, oxygen demand keeps increasing, but they're unable to deliver the appropriate amount of oxygen to tissues that need that oxygen, and so performance crashes when we have this mismatch between oxygen demand and supply. Now, note here that this is a continuum and that these two hypotheses are not mutually exclusive. A recent debate in the literature, and this is actually like really heated debate, like people were yelling at each other in meetings a few years ago and stuff about this. Doesn't have to be one or the other, is that both of these hypotheses can prove true in different situations. But as a graduate student, I was really fixated on this oxygen limitation hypothesis. It had barely been tested in terrestrial ectotherms, primarily work had been only done in fish. And so we set out to test this mechanism for setting upper thermal limits. And the study I'm going to show you here was one of the studies that was part of my dissertation here at ISU. And you can see a little bit of foreshadowing about the result of that. Chapter 3 is titled, But We Don't. So this study utilizes the western terrestrial garter snake. This is a species found across much of the western US and southwestern Canada. It's a small, common species across much of its broad geographic range. And our study sought to test the mechanism setting the high temperature limit in this species. So we performed an experiment at which we measured we measured the organismal response across a range of temperatures. This represents the active range of the animal right up to temperatures that are just below what would kill the animal under that amount of exposure. We tested this oxygen limitation in this terrestrial tetrapod, and in this test, we characterized both the hormonal and the energetic response to these high temperatures. So we measured oxygen consumption as a measure of metabolic rate. We measured glucose to indicate the amount of available energy substrate. We measured the hormone corticosterone as an important hormone regulating energy balance generally and often implicated in the stress response specifically. And we measure the hormone insulin, which is important in regulating glucose metabolism. And this work was done with my fantastic advisor at Iowa State University, Ann Bronikowski, now at the University of Michigan, as well as fellow graduate students Rory Telemico, who's now at Fresno State, and Caitlin Holden, who is still here as a postdoc at Iowa State University. And so here is what we found. First, in terms of what I'll call the stress response, for all the figures I'm going to show here, we have the body temperature across the x-axis, and we have our dependent variable on the y-axis. Here, the measure of corticosterone concentration in the blood plasma. And you can see a very strong effective temperature. Cort just skyrockets at this high temperature. And this indicates that these high temperatures are in fact stressful to these organisms. At the same time, we measured the amount of available glucose you see here, and you see a very similar response. However, the shape of this curve is a little bit different in that the glucose is elevated at 35 degrees, much higher than corticosterone is relative to baseline levels. And this suggests that even though cort and glucose are both responding to temperature, that perhaps it's not just corticosterone increase that's driving this increase in glucose. In terms of energy regulation, we also found that insulin increased across the range of temperature here. And this is important. This is actually the first study to show that insulin is temperature dependent in an ectothermic tetrapod. And finally, we found that metabolic rate increased rather dramatically at the highest temperature as well. And this is our important result to test the hypothesis of oxygen limitation. You can see here that at the highest temperature that there is no reduction in oxygen consumption. And so even at these very close to near lethal temperatures, these snakes are not suffering from a reduced capacity to deliver oxygen, but rather other factors. So we found very robust results here that do not support this oxygen limitation hypothesis. We found no support for this hypothesis. I was a little bit heartbroken as a graduate student. I really wanted this result. Could convince myself before we did the experiment that this would be our result, but in fact, we did not find support for that. Instead, we found that extreme temperatures induce interesting energetic and hormonal responses, and that this response is outside of the normal homeostatic range for these organisms and likely has long-term effects on energy balance, which is important, but doesn't necessarily address the question of what's the proximal factor that drives this reduction in performance at these high temperatures. So we didn't find support for that experiment, so we do what any good scientists do. We try something else. We try to explain results using what am I trying to say there? We try another approach. And for this, I want to return back to our thermal performance curve. And you can see a curve here on the left. And what's important to note is that these curves can shift under a variety of ecological conditions. They can shift across ontogeny. So as an organism grows, the shape of that curve can shift. And they can also shift in a variety of different ways. So the curve can change its position. The optimal temperature can change even as the critical thermal minimum and maximum stay where they are. The entire curve can shift left or right. And of course, it can become broader or narrower, representing a more generalist versus a more specialist in terms of thermal physiology. So with Rory Telemico and I worked to try to explain very disparate results in the amphibian and reptile literature in a common framework. And we came up with something we call the hierarchical mechanisms of thermal limitation hypothesis. So let me walk you through this. So this figure here shows, again, body temperature on the x-axis, and on the y-axis shows oxygen use or demand. And the blue line here is the resting metabolic rate. We fit this line to published data on pythons. And this is the temperature dependence of the resting metabolic rate in pythons. The red line represents the maximal metabolic rate, the maximal metabolic capacity across a range of temperatures in those same pythons. And in pythons, to elicit that maximal metabolic response, they fed them a large meal. Pythons are sporadic eaters, and when they do eat, it incurs a great metabolic cost to digest that meal. So they induce this maximal metabolism by feeding the pythons a big meal. And what's important here is you can see that these lines have a different shape, but that they converge out here on the right-hand side of the figure. What's my next slide here? Oh, actually, before I get to that, what's important here too is the difference between this resting and maximal metabolic rate. That difference between those two lines represents something called aerobic scope for activity. Aerobic scope for activity is essentially the amount of aerobic capacity that an organism has to fuel long-term activities. So how much can it up its metabolic rate from resting to maximum to fuel things that it needs to do in the long term? You can see that far out on the right here, the red and blue lines converge, representing a temperature at which this aerobic scope for activity is essentially zero. So at this high temperature, this organism has no aerobic scope to fuel long-term activities. Also important is to note this black vertical line here represents the the upper critical limit of this organism. And this is from other studies in this python species. And notice here that this subcellular T-crit is to the left of the aerobic T-crit, meaning that as temperatures increase, this organism's function is going to crash at a lower temperature than the temperature at which that aerobic scope becomes zero. Now, under conditions of oxygen limitation, we can predict how these relationships will change. And specifically, we see that if oxygen is limited under conditions of mild hypoxia, this aerobic scope for activity decreases across the range of temperatures, and this aerobic T-crit shifts to the left a little bit. Now, important to note that it's shifted to the left, but it's still to the right of that subcellular T-crit. So the critical thermal maximum isn't shifting, even as the shape of these curves overall change. Now, I mentioned that we're using aerobic scope for activity as an important measure of performance, and that's because aerobic metabolism fuels essential long-term activities that are tied to fitness. So this includes things like digestion, growth, and reproduction. So we can use aerobic scope as a proxy for these important fitness-related processes. Now, if we take the changes in the curve that I just showed you and bring it back to our thermal performance curve, we have something that looks like this. Under conditions of normoxia, we have this classic shape of the thermal performance curve where, where temperatures rise gradually to the optimum temperature for performance and then decrease down to a critical thermal maximum. Under these conditions of mild hypoxia, mild oxygen limitation, the curve shifts predictably in that the maximum performance is reduced, it's downshift, and also this optimal temperature for performance shifts a little bit lower as well. Now, importantly, under this level of oxygen limitation, we don't see any change in that critical thermal maximum because that aerobic scope doesn't become zero until a temperature that's above the maximum, the critical thermal maximum set by these subcellular processes. Now, if we return back to our graph here showing aerobic scope for activity between resting and maximum metabolic rates, and we subject this to extreme hypoxia, we get something that looks like this. In this case, that aerobic scope is dramatically reduced, and importantly, that aerobic T-crit, where those blue and red lines intersect, is now shifted to the left of the critical thermal limit set by subcellular processes. So in this case, we might expect a thermal performance curve again to change in predictable ways that look like this. Under extreme hypoxia, that maximum performance is dramatically reduced and more further left shifted. And under this condition, the critical thermal maximum can also be reduced. Now, we developed this framework based on our extensive review of the literature of thermal performance in reptiles and amphibians that provided some kind of seemingly contradictory results in how thermal performance curves change over a range of temperatures and the conditions in which these different processes are important. We synthesized that, and under this hypothesis, we can form some key predictions. The first is that thermal performance curve changes shape with reduced oxygen capacity in predictable ways, specifically that peak performance is reduced, the optimal temperature is reduced, and the thermal performance curve, this results in a thermal performance curve that's more symmetrical in shape. Importantly, also under conditions of mild hypoxia, that critical thermal maximum is not changed, and that's in fact the most robust component of the thermal performance curve. So we developed this framework, we proposed at how that might work, and then, as we do as researchers, we went out and tested this idea. And this brings us to chapter 5, and maybe it's right. So the work I want to show you for this chapter, it emerges from my postdoc with the CNRS in France, and specifically Project Podarcis, which, as you're about to see, is not only the name of the genus in which this beautiful lizard is found, but also the world's greatest acronym, standing for Potential Oxygen Limitation of Distributions and Responses to Changing Climates in Ectotherms. So with this postdoc, we conducted a series of experiments with the common wall lizard, Podarcis moralis. These guys have a broad range across much of Western and Southern Europe. You see on the range map here. They've also become established in a number of other places throughout the world as well. And across this broad geographic range, they inhabit a variety of habitats, ranging in elevation. They live in cities, they live in more naturalistic landscapes, but generally, they live at lower elevations. Now, in the Pyrenees Mountains in the south of France, where we conducted this work, in recent years, temperatures have increased, and these guys have been documented to move up in elevation along this elevational climb as those habitats become thermally suitable. So we wanted to understand how this new environment, specifically the oxygen limitation found at higher elevations, would affect these lizards, and then be able to predict if they would be able to continue moving up in elevation as those habitats continue to warm as they will. So I want to show you some results from those experiments, but I want to take a brief aside here for a second, because I'm going to show you some really nice looking graphs in just a moment that show some clean results and tell a clear story. But I want to just give an aside, especially for those of you that are doing any experiments with animals, and talk about how we got there. And for this, I'm going to bring it way back to Herman Melville, 1853 story called Bartleby the Scrivener. And this is one of my favorite short stories. And it tells the story of a clerk working in a law office who one day decides that he would rather not work. He doesn't quit his job. He doesn't go away. He goes to work. And when he's asked to do something, he just says, I'd prefer not to. They ask him to do something else. He says, I'd prefer not to. Well, I like to think of our wall lizards as very much like Bartleby. They very much preferred not to participate in our experiments. They're a little more aggressive than Bartleby. You see an example of one biting there. You see this guy somehow miraculously tied a knot, or more than a giant mess of a knot. We see one here biting its own tail. It's unhappy with its thermocouple. And another one here exploring Brooke's hair. Even that was as we tried to release it back into its habitat. It was still like, no. This guy, I don't know what the heck that guy's doing. And sometimes they would get loose in the lab and it would make us do crazy things and get very excited when we'd finally found them. So I just want to give a little bit behind the scenes there, that even though we have these nice clean results, there's a lot of chaos that went into conducting these experiments. So our first set of experiments utilized transplant experiments, and we brought these wall lizards up to high elevation to test a variety of organismal responses. I want to just summarize a few of those results here. The first is from an experiment in which we measured the maximum metabolic rate of these lizards. And you can see on this figure, we measured this at 3 time points across the time of the experiment. On the y-axis there is their maximum metabolic rate they attained. You can see the red lines represent lizards kept at low elevation, so at their native elevation, on conditions of normoxia, whereas the blue line represents animals brought to high elevation under conditions of hypoxia. And you can see that across all of our time points, there's a very clear signature that hypoxia was limiting their maximal metabolic performance. This had long-term energetic consequences for the lizards as well. This figure shows body condition on the y-axis across our 3 time points of the experiment. And you can see by 6 weeks, there was a very dramatic difference in body condition, or the mass relative to the size of lizards, whereby lizards at high elevation were losing body mass, presumably because of their aerobic limitation in long-term activity over the long term of time at high elevation. In other words, they could survive at high elevation for a short time, but over the long term, they didn't do so well. We conducted a similar experiment transplanting the eggs of the snake Natrix mora to incubate under different temperatures and at different elevations. You can see one of our results here. The y-axis shows the size of offspring at hatching. And the takeaway message here is that when you combine bringing an animal to high elevation with reduced oxygen availability and incubating at high temperatures, we find a dramatic reduction in body mass at hatching compared to any of the other conditions. And this is important because body mass at hatching for a snake has important fitness consequences. Many studies have shown that larger snakes that hatch larger survive better in the wild. And so the combination of high temperature and low oxygen were detrimental to the development and growth of these guys. And that fits with our understanding of how temperature and oxygen are going to interact. So these are some interesting results from some of our first studies, but we wanted to go in and test the hypotheses of the hierarchical mechanisms of thermal limitation more explicitly. And to do this, we conducted another transplant experiment in which we estimated the thermal performance curve across 7 temperatures at 2 different elevations for 2 different performance measures. And this is again in the common wall lizard. We measured them at their elevation of capture at about 400 meters above sea level, and then we transplanted them up just above, a couple hundred meters above their current elevational range limit. And this work was in collaboration with Roy Talamico again, and also Antonio Cordero, proud graduate of the EEB program, who's now at the University of Lisbon. So this is the view from the high mountain station where we transplanted lizards. This is the Pyrenees Mountains. Somewhere in there is the boundary between France and Spain. Beautiful location to do work, but we were completely engrossed in our experiment. In fact, this is one of the most insane experiments I've ever conducted, given the time limit we had to do it. You can see here collaborator Essie Rogers with some of our homemade plumbing from our respirometry equipment. We used 3 respirometry machines at the same time, a set of laptops. It was absolutely nuts. But I'm glad to say we had a good time. And thanks to the friendships that I forged at ISU, we didn't get too crazy. We had a really nice time working on this project together. That's me and Rory and Antonio. We're close. So what did we measure? Our first measure of performance was sprint speed. Oops, there we go. And sprint speed is a commonly used metric of performance in lizards, has important implications for predator evasion as well as prey capture. And so we measured sprint speed in these animals at low and high elevation. Now it's important to note that sprint speed is the vast majority of power for sprint speed comes from anaerobic metabolism. So we didn't expect sprint speed to necessarily be limited by oxygen in the short term. If we bring this back to our measures, our figure here showing performance measures. Oops. Actually, let me make that transition again. So that's our first measure of performance, sprint speed. For our second measure of performance, we measured aerobic scope. So to bring this back to our figure, remember aerobic scope is the difference between the resting metabolic rate and the maximal metabolic rate. And it's important for fitness, long-term fitness-related processes. Now, to measure this in animals is not always easy because in the same individual, you need to get a resting metabolic rate and a maximum metabolic rate. It can take a lot of time. We developed some novel methodology to do this using these little remote-controlled robot insects to elicit the maximal metabolic response from these animals. Let me show you a video of what that looks like here. And you can see we have the animal in its metabolic chamber, and we can drive these remote-controlled insects around and perturb the animal and elicit this maximal metabolic response. And we actually have another paper in review right now in which we show that the aerobic response elicited by this technique is greater than that of traditional techniques such as running an animal to exhaustion and then measuring its aerobic metabolic rate. I want to note that we checked this very carefully. There was no external injuries on any of these animals. And in fact, we paired animals so that we tested sprint speed on animals subjected to this and not subjected to this. And there was no difference in sprint speed immediately after this treatment. So demonstrating that even though we're eliciting this maximal metabolic response, we're not causing any long-term injury to the animals. And so to explain this, I'll show you a figure here. This figure shows you a time in minutes of our measuring our metabolic rate. The y-axis shows the oxygen that is consumed by an animal. So we have the animal in the metabolic chamber. We measure its resting metabolic rate for a time. And then we begin perturbing the animal with this hex bug. And within about 5 or 6 minutes, this elicits this maximal metabolic response. We then, using the resting and maximum metabolic rates, we get a measure of aerobic scope for each animal. Great. So now we've got this experiment. We measured these 2 performance traits at low and high elevation. We want to test our ideas. And to do that, we brought things back into the thermal performance curve paradigm. Specifically, we created thermal performance curves by fitting what's called the beta function. The beta function is a way to parameterize these curves. And what's important about this function is that it's been demonstrated to fit well with lizard performance generally and ectotherm performance generally and lizards specifically. But also appealing to us is that it includes some rather intuitive parameters. The first is the measure of optimal, the maximum performance, the optimal temperature for performance, and the critical thermal maximum. So these performance have direct biological relevance, which was important to us in the questions we were asking. And specifically, we predicted that hypoxia exposure would reduce this maximum performance, would reduce the optimal temperature for that performance, and furthermore, would not affect the critical thermal maximum. So we wanted to parameterize our curves for that to test how this oxygen limitation would affect these different measures of performance. Now, we didn't measure critical thermal maximum in this experiment, but fortunately, another experiment we did, this is another collaboration with Brooke Bodensteiner, and you can see here, and bringing animals up to this high elevation caused no effect on their critical thermal maximum. And so we use this empirical result to parameterize our model and set our critical thermal maximum to the average from that study, which is just over 41 degrees Celsius. So we measured these, we created these curves. Let me show you what they look like. First, our thermal performance curve for sprint speed. So you see sprint speed on the y-axis here, the range of body temperatures on the x-axis. You see this very classic thermal performance shaped curve. And importantly, you see no difference in those curves in lizards measured at low versus high elevation. So as we predicted, this oxygen limitation does not affect the sprint speed because that sprint speed is primarily anaerobically fueled. Now, if we look at the results for aerobic scope, we see a much more dramatic difference in that animals kept at low elevation achieved a higher aerobic scope, and they did so at a higher temperature compared to animals that were subjected to this high elevation hypoxia. And you can see that nicely right here. And I want to take a second and just revel in this figure, never before and maybe never again, while I have an experiment where the empirical results so neatly match our theoretical expectations. We're very happy with this. And so now we have this. We wanted to take this one step further. The utility of such a model comes when we can use it to form predictions about how these novel environments will interact to shape organismal performance across these different axes of variation, specifically here, temperature and oxygen. So we created something called a temperature oxygen performance surface, or TOPS. To do this, we took the beta function and expanded it with two additional parameters that describe the relative, we took the beta function, and first we added a parameter that describes the relative oxygen availability, simply the amount of oxygen available relative to reference conditions, here relative to sea level. And then we included two parameters that describe the sensitivity of performance to performance to variation in oxygen. So specifically here we have a gamma, which describes what's going on there. What are you doing, buddy? Oops, let me go back. Gamma, which describes the sensitivity of performance to oxygen availability, and tau, which describes the sensitivity of temperature. And so we can put this together for sprint speed and aerobic scope, and you can see what this looks like here. And again, matching our theoretical predictions, you can see that sprint speed is not sensitive to variation in oxygen, whereas aerobic scope very much is. And this is borne out in our parameter estimates. You can see this parameter estimate for gamma, which is the sensitivity of performance to oxygen availability, is much higher in aerobic scope than sprint speed. So to go back to our key predictions of the hierarchical mechanisms of thermal limitation hypothesis, all of these predictions were met. We found that thermal performance curve changed predictably. The critical thermal maximum was unchanged under this mild hypoxia, while at the same time peak performance and the optimal temperature for performance were reduced. And so we found pretty robust support for our expectations. This is great. We plan on expanding this. We have some work in progress right now, expanding this to other taxa, expanding the range of oxygen and temperature conditions under which we're measuring this performance. Great. Now, if we return to our table of contents, we get to the last chapter, but probably not always. So everything I've shown you so far is our expectation and empirical results relating to organisms that generally live under high oxygen availability at low elevation. What happens when we subject them to reduced oxygen availability at high elevation, right? What happens if you take something used to a lot of oxygen and reduce the amount of oxygen available? What if, on the other hand, we have organisms that are used to living at high elevation where there's not much oxygen, and we bring them down to a more oxygen-rich environment? And that's exactly what we did in an experiment with this lizard, Iberolacerta benali. So these are high elevation endemic lizards. There's 3 species in this genus, native to the Pyrenees Mountains, and they're all critically listed due to the effects of climate change, but also potentially through competition as more general species like Podarcis moralis move up in elevation. Now with these guys, we conducted a transplant experiment in exactly the opposite direction. We brought animals that are endemic to high elevations and brought them down to low elevation. And you can see here our results for the preferred body temperature. So this is the temperature an animal selects in a gradient. And you can see that the low elevation animals actually selected a lower body temperature compared to high elevation animals. Now this is exactly the opposite direction that we would expect if the same mechanisms were in place when low elevation organisms are brought up to higher elevation. And you can see it's a pretty dramatic reduction there, almost 3 degrees Celsius lower body temperature they selected. We also examined sprint speed, and we found that at low temperatures, you see on the x-axis here at 15 and 22 degrees Celsius, the maximum sprint speed did not differ between animals at their native high elevation and at low elevation. But at higher temperatures, we found a significant difference. with sprint speed being reduced at the low elevation, the high oxygen concentration. Again, directly contrary to what we might predict, even though sprint speed is anaerobically fueled. And if I show these data another way, you can see a very clear divergence at these high temperatures where the transplanted animals are running much slower relatively. So what is going on here? The short answer is we don't know. The longer, more interesting answer is that it probably has something to do with aerobic metabolism in the mitochondria. So recent review has demonstrated, along with a huge body of research, the importance for the mitochondria, the organelle, commonly known as the powerhouse of the cell. This organelle is essential for organismal energetics in balancing the simultaneous demands of aerobic capacity, so fueling activities, with the production of producing potentially harmful side products in reactive oxygen species. And the mitochondria is constantly in this balance between providing fuel while at the same time not producing these damaging molecules. And this can shift in a number of important contexts. So we're not sure if this is what's causing this reduction in performance under high oxygen concentration, but this is an avenue that we think might be useful to explore. And the reason we think it might be useful to explore is because a number of other studies have shown that the mitochondria can play a key role. And I want to present one last case study here. I'm going to bring us back to garter snakes, where we started, and present one last study. And this study is in collaboration with Tanya Schwartz, who's now faculty at Auburn University and a proud alum of the Bronikowski lab here at ISU as well. And in this study, we found, actually, let me go here. I kind of skipped ahead here. I'm going to just go back to this before I talk about Tanya. What we found in this study is that a single nucleotide polymorphism, a SNP in the mitochondrial genome, segregates predictably across the landscape where we find these animals, these garter snakes. So you can see here the relative ratio of the two alleles in yellow and green, and you can see that it's much more prevalent in some populations compared to others. And this distribution fits nicely, where do I want to say here? I might change the order of these slides a little bit. This distribution fits nicely with our understanding of this ecotypic variation in these snakes, whereas these lakeshore populations have a fast pace of life. They grow really big, produce large litters, but they die at a young age, relative to snakes living in the mountain meadows, which have a much longer lifespan, reproduce more intermittently, and have smaller litters altogether. And so you can see here that these mitochondrial alleles segregate neatly across the landscape in conjunction with these life history ecotypes. Now, importantly, this SNP actually codes for a section, or for a protein, for part of a protein, I should say. What do I want to say? This SNP affects the amino acid that's incorporated into a protein that's part of complex 3 in the electron transport chain of the mitochondria. So we have a change from the ancestral state of a thymine to a cytosine, which changes the amino acid from isoleucine to threonine, which actually changes the polarity of that protein and therefore affects the action of the electron transport chain, which, as you know, is where aerobic metabolism happens. And if we look at the results of this on an organismal level, we find that there are sex-dependent variation in whole organism metabolic rate that depends on which of these SNPs is present. In other words, the SNP affects the metabolism of females differentially from males. Now, we did a little bit more with this in the study, but the takeaway message here is that this variation at the molecular level affects whole organism energetics in a way that fits with our understanding of variation across populations and across the landscape. And so with this paper, we took these results along with some previous results from studies in this system, and we came up with this generalized framework describing the effects of environment and genetic background on oxidative phosphorylation and how that in turn affects reactive oxygen species production and mitigation, how that affects whole organism performance, and how that plays into organismal traits like life histories and aging, so reproduction and aging. And so all of this work here brought us to this more generalizable framework which I'm interested in bringing back to our lizards in the mountains and testing these ideas to better understand how temperature and oxygen interact, how that affects organismal performance, and how ultimately that will affect the ability of these organisms to live in these novel environments. So having this new framework that we want to test, this brings us right back to chapter 2. We think we might understand how. And so the cycle of continuing to develop and test these hypotheses continues. And we're right back to where we started, trying to understand things and learning along the way. And that's all I got. Yay. Cool. So that was about 40 minutes.
40 minutes exactly.
Yeah, okay, that's actually perfect. That's good. So that's just, thanks for being a good audience. I know it's a little long, but it's kind of exciting to put all of this work that I've done over the past, I don't know how many years, 10 years, into like one kind of coherent story. So thank you. With that in mind, we're getting close to closing time here. Any last comments, thoughts, suggestions on what's going on in class? Oops, as soon as PowerPoint will cooperate, I've got a couple last slides with some reminders. And actually, Princeton, can I ask you a favor? Actually, don't hand these out yet. I want to show the class averages yet first.
I got my girlfriend right presentation.
Yeah, go ahead, Ethan.
How did you measure aerobic consumption or
Oh, gas analyzers. So we can measure the relative concentration of CO2 and oxygen.
Okay. So just like metabolism then?
It's a proxy for metabolism. Yep. Basically, it's a proxy for aerobic metabolism anyway.
So then you basically measuring that at the resting level?
And then at the maximal level with those hex bugs.
Okay.
Yep. All right, so where is our presentation for class? Okay, so as promised, I have your quizzes graded. Here's a histogram of results. Again, this shows how many people got a different score. The y-axis shows, or x-axis shows the score, y-axis shows the number of people. So, in other words, 7 of you got between, I don't know where the division is here, somewhere between a 17 and a 20. About 6 of you got between a 16 and a 17 or so. The class mean was 16, which is right about 80% on the quiz grade. And we can actually, Princeton can pass those back right now. I'm happy to answer any questions. I gave you detailed comments, or at least some comments if you didn't get full credit on a question. If you have any questions, obviously just let me know. The last question, thank you, did have 2 correct answers. And so what I did for that is there were 4 options, and basically each of those counted for half a point. So if you didn't select one that was not supposed to be selected, you got half a point, and so on. And so that question, you get some like fractional points to make it fair. We had a couple 20s in the class. Nice job. Kudos to those of you who got it. Take a look at the distribution. Just know if you're falling out on this side of things. Might want to check in with me, change your study habits a little bit, be better prepared for quizzes in the future. Speaking of which, lecture exam number 1 is not too far off. It's going to be in class on Wednesday, March 16th. This is some new information for you here. It's going to be closed book and closed note. You can expect a combination of multiple choice and long answer questions. It's going to cover everything that we've done up through today. It's a lot. I'm writing the exam with the intention it'll take about 50 minutes. You'll have 70 minutes to take it. Those of you with extended time will have an extra 35 minutes, so you have 105 minutes to take it. We can work on those details as we get closer. Remember, our first class back is going to be a review session. I don't want to just throw this at you after spring break. It will be helpful if you can bring some questions to that review session, stuff you want to go over, talk about, review in advance of the exam. My recommendation, each of your class programs has learning outcomes. And my recommendation is to go through and use those as a study guide. All of the questions will come from those learning outcomes. So make sure you review those. We've got lots of lecture materials. We've done a lot of other activities with podcasts and videos and everything like that. So make sure that you're brushing up on all of that. I realize it's a lot of material, and that's why we're having the review session right when you come back. Got one last slide here with a couple other pieces of information, but any questions you have about this? Okay, last slide here, that's to send you off. There's no class program. Personally, my philosophy is that spring break should be kind of a break from class for you. Take a breather, make sure you're feeling good, do all that stuff. I know I'm looking forward to it. So no work, no assignments, no class program over break. And then after spring break, just remember, we've got the review session the first day back, and then we'll have that exam on the 16th. Also, tentatively, oh, this isn't on the slide. The following Wednesday, the 23rd, I've tentatively rescheduled the mud puppy trip. So I'll give you those details when we get back, but just know that we're going to try that for the 4th friggin' time. And eventually we're going to see some gosh darn mud puppies this semester. Let me know if you have questions about anything. Otherwise, I hope everyone has a really fun / relaxing / productive / whatever you want it to be spring break. Thanks, everyone.